gene therapy. So gene therapy, we will start with gene therapy. An introduction to genetics in human diseases. About 4,000 human diseases are thought to be inherited, which means around 4,000 different diseases are thought to be from the parents. So when the parents have, it is inherited to the offspring. So it is passed on to the offspring. So about 4,000 human diseases are thought to be inherited. Scientists are making good progress figuring out where genes are located on chromosomes. Genetic diseases are caused by either mutations or incorrect sequences in the normal form of the gene. So imagine a situation, you have a gene. In case if the normal form of the genetic material is converted to abnormal form by either mutation or by means of deletion, you get a genetic disorder. Or when it is replaced by an incorrect sequence, you will get a genetic disorder or genetic diseases. I repeat, about 4,000 human diseases are thought to be inherited. Scientists are making good progress figuring out where genes are located on chromosomes. Genetic diseases are caused by mutations or incorrect sequences in the normal form of the gene. What is gene therapy? A very important 2-mark definition. So gene therapy is the name given to the methods that aim to cure an inherited disease by providing the patient with the correct copy of the defective gene. Imagine a situation, a person has a defective gene, you're removing the gene and replacing it with the correct version. And the methods involved in doing so is referred to as gene therapy. I repeat the definition. Gene therapy is the name given to the methods that aim to cure an inherited disease by providing the patient with the correct copy of the defective gene. So what are the types of gene therapy? Gene therapy basically are of 2 different types. One is gene replacement therapy, which is also referred to as gene augmentation. Again, these are very important 2-mark questions. Gene replacement therapy, also referred as gene augmentation, and the other one we have is corrective gene therapy. What is gene replacement therapy or gene augmentation? Random insertion of healthy counterpart of defective gene somewhere in genome so that its product could be available. Imagine you have a stretch of DNA molecule, you have a defective gene in the center. You need not replace the correct version in the center. Any part of the genome you can replace so that the product is obtained. And such therapy is called as gene replacement therapy. I repeat, random insertion of healthy counterpart of defective gene somewhere in genome so that its product could be available. And it is also referred to as gene augmentation. On the other hand, we have corrective gene therapy, directing insertion of healthy gene at a specific site to displace defective gene. Imagine a situation, again you have a stretch of DNA molecule or a gene. In the center you have a defective gene. You are removing the defective gene from the center and replacing the correct version in the same place, in the center. And that type is called as corrective gene therapy. I repeat the slide again. Gene replacement therapy, random insertion of healthy counterpart of defective gene somewhere in genome so that its product could be available. It is also referred to as gene augmentation, corrective gene therapy, directing insertion of healthy gene at a specific site to displace defective gene. There are 2 approaches to gene therapy. The first approach we have is germline therapy and the other one we have is somatic cell therapy. What is germline therapy? A fertilized egg is provided with a copy of the correct version of the relevant gene and re-implanted into the mother. Imagine a situation, a mother is having a newborn baby that has a defective gene. During her pregnancy, the egg is removed from the mother, the correct version is inserted and re-implanted in the mother is called as germline therapy. I repeat, a fertilized egg is provided with a copy of the correct version of the relevant gene and re-implanted into the mother. If successful, the gene is present and expressed in all cells of the resulting individual. Germline therapy usually is carried out by micro-injection of DNA into the isolated egg cell and theoretically could be used to treat any inherited disease. I repeat the slide again. Germline therapy. A fertilized egg is provided with a copy of the correct version of the relevant gene and re-implanted into the mother. If successful, the gene is present and expressed in all cells of the resulting individual. Germline therapy usually is carried out by micro-injection of DNA into the isolated egg cell and of course is theoretically be used to treat any type of inherited diseases. So you have seen this already in previous slide when I taught you like what is called as tissue plasminogen activator reduction. So micro-injection is one technique where you use a fine micropipette to inject the DNA in the host nucleus with an holding pipette. Next one we have is somatic cell therapy, which is the second approach of gene therapy. Somatic cell therapy involves manipulation of ordinary cells, usually ones which can be removed from the organisms, transfected and placed back in the body. Imagine a situation, you have some blood disorder. You're removing blood cell, you're replacing the original form or the correct form, inserting the blood cell to the place where it was found earlier. That is what is somatic cell therapy. I repeat, somatic cell therapy involves manipulation of ordinary cells, usually ones which can be removed from the organisms, transfected and placed back in the body. This technique is most promising to cure inherited blood diseases. Example, I have given you two examples. The first example is hemophilia, where the blood clots very slowly. And the second disorder which I have given you is, as an example, is thalassemia. It is a type of anemia found in Mediterranean countries due to the defect in the production of hemoglobin. So there will be less production of hemoglobin in thalassemia patients. So I repeat the slide. Somatic cell therapy involves manipulation of ordinary cells, usually ones which can be removed from the organisms, transfected and placed back in the body. This technique is most promising to cure inherited blood diseases. Example, hemophilia, blood clots very slowly, and thalassemia, type of anemia found in Mediterranean countries due to a defect in the production of hemoglobin. So this slide shows you a picture of hemophilia. It is a blood disorder in which a person's blood does not clot properly. It is a recessive sex-linked X chromosome disorder. So if you have a defect in X chromosome, the disease is inherited. And 1 in every 10,000 males born are usually affected. And of course, this is called as a royalty disease. So if you see on the left picture, you have a mother who is a carrier of infection and the father is a normal person. Mother will have an double X chromosome, one chromosome a normal X and the other X, if you see, has a defective slash on it. And on the right hand side, the father normal will have XY. In the mother carrier, if you see, one X is replaced by a hemophilia gene in it. So when there is crossing over or cross combination occurring between the mother and father, you have these combinations, XX, XY, X-X and X-Y you will have. The XSX which is generated will be a daughter who's normal. A normal XY combination will be a son who's normal, whereas X dash which has hemophilia gene with a normal X gene will be a daughter who is a carrier. And of course, the other one, the XY, X-Y, who will be a son with an hemophilic disorder. That is how it is going. Why is usually a mother carrier here in this case, if you see, is one X is defective, even though she can compensate with a dominating X from an other gene. So if you see, double X is there, so she always remains as a carrier and infects the persons who are inherited from her. So that is how it goes in the case of hemophilia. I repeat again, a disease in which a person's blood does not clot properly. It is a recessive sex-linked X chromosome disorder. 1 in every 10,000 males born are affected. And of course, the disease is also referred to as a royalty disease. And this is sickle cell anemia where you have recessive allele. If you look into the picture, the red blood cells, normal is spherical in shape. And the red blood cells that is undergoing sickle shape is causing anemia and of course pain when it is circulated in the blood. So again, this order can also be treated with help of somatic cell therapy. Red blood cells are sickle shaped, issues with circulation causing anemia and pain. What are the cellular targets of somatic gene therapy? Gene therapy as such is a very important 5-mark and a 10-mark question, plus of 2 marks importance. So cellular targets for somatic gene therapy. As I told you, somatic gene therapy does not involve the egg from a surrogate mother or egg. It involves body cells. So what are the different cell types that remain as a cellular target for this therapy? If you see, are bone marrow totipotent stem cells, hepatic cells, fibroblast cells, endothelium and pulmonary epithelial cells, endocrine cells, cells of central nervous system, smooth muscle cells of blood vessel walls. I repeat again, cellular targets for somatic gene therapy. Following cellular targets have been explored for gene therapy, that is somatic gene therapy: bone marrow, totipotent stem cells, hepatic cells, fibroblast cells, endothelium and pulmonary epithelium cells, endocrine cells, cells of central nervous system, smooth muscle cells of blood vessel walls. So what are the common methods used for gene transfer? We have 4 different methods which is used: microinjection, electroporation, use of viruses as vectors, an injection of liposomes, among which the common ones that have been used recently, if you see, are the microinjection technique and use of viruses as vectors. So 2 techniques which is predominantly used for gene transferring mechanism in gene therapy, if you see, is microinjection and uses of viruses as vectors. Even then it has 4 methods in gene transfer: microinjection, electroporation, use of viruses as vector, and injection of liposomes. This picture will give you a basic idea of liposomal attraction or liposomal transfer. You have in this left hand side a white color picture in which you have the center one, the circular one, lipid bilayer, which is having an hydrophobic and hydrophilic end. So rend attach ana madri irukku on the left hand side in the center circle. So with DNA, which remains as a foreign, it gets attracted to the liposomes. That is why it is called as liposomal attraction. On the right hand side, it is an original picture of how liposomes come in contact within cell. Gene therapy. We are coming inside the topic very sincerely. To design and carry out a gene therapy treatment, what do a researcher need to have or should do? To design and carry out a gene therapy treatment, a researcher must identify the gene or genes responsible for the disorder, make copies of the normal gene, insert the copies into the vectors, infect the affected cells with the vectors, activate the gene so that transcription and translation takes place. First, to identify a genetic disorder, he should be responsible to know what gene is coding for what protein molecule or what enzyme molecule that it codes for. So the first predominant function for a genetic researcher, if you see, for gene therapy, he should identify the gene or genes responsible for the disorder. So you know what is a defective gene, what is the original form of gene. The next step is making copies of the normal gene. So one gene inserting in a one vector and getting 100% successful is no way possible in genetic engineering. You have to synthesize a normal gene in larger proportion or in larger number. So you have to make copies of the normal gene, and you need a vector system to insert because the naked DNA usually gets dissolved or dissociated. So vector is always important in order to multiply or prepare copies of the DNA. So insert the copies into a vector. After inserting, you have to transfect, infect the affected cells with the vector, and of course you should activate the genes so that transcription and translation takes place. I repeat the slide. To design and carry out a gene therapy treatment, a researcher must identify the genes responsible for the disorder, make copies of the normal gene, insert the copies into vectors, infect the affected cells with the vectors, and activate the genes so that transcription and translation takes place. So this picture shows you how is it done. Look into the picture. The top yellow one, if you see, is a viral vector carrying the healthy gene. So top left, if you see, you have an yellow color picture with a red color double-stranded DNA within it. So it is a vector, viral vector carrying the healthy gene. Below that, you have 3 different circles depicting the cell. So the first cell on the left-hand side is the cell with mutated genes. So mutated portion is marked with blue in double-stranded DNA molecule on the left-hand side picture. So cell with mutated genes. In the middle picture, if you see, the viral vector is transfected or attached to the cell so that the healthy counterpart or the healthy gene is inserted inside the cell. So the second picture from the left, vector inserts healthy gene into the cell. If you look into the picture, you have red colored double-stranded DNA molecule from the viral vector being inserted in the cell which has a mutated gene. And the third one, if you see, the third side from the left, and the first picture from the right, if you see, new gene in the cell along with the original gene. So already the cell has a mutated gene, you're inserting a new gene, so automatically the new gene will start functioning. Functional proteins are created from the therapeutic gene, causing the cell to return to its normal state. So this is what is the picture. So how is it done? Cells with mutated gene, you have vector inserts healthy gene into cell. So you will have a viral vector carrying the healthy gene, a new gene in the cell along with the original genes is formed. And the functional proteins are created from the therapeutic gene, causing the cell to return to a normal state. Viruses as vector. So gene therapy mostly involves, like for example, what you call as germline therapy, we go with what is called as micro-injection technique, as I told you earlier. But for the other somatic cell or a normal cell gene therapy, if you see, you commonly prefer viruses as vectors. Viruses, they replicate by inserting their DNA into a host genome. So if you see, they are obligate intracellular parasites. It requires a host cell for its replication or multiplication. Gene therapy can use this to insert genes that encode for a desired protein to create the desired trait. So that is the idea behind using viruses. Viruses, if you transfer a gene in a virus, when you put that virus with a cell, it will automatically transfer or infect the cells and inserts a DNA inside the cell. That is what is the idea behind. So I repeat, viruses replicate by inserting their DNA into a host cell. Gene therapy can use this to insert genes that encode for a desired protein to create the desired trait. There are 4 different types of vectors, viruses that are used as vector molecule. What are they? If you see, adenoviruses as vectors, adeno-associated virus as vectors, retrovirus vectors, and herpes simplex virus as vector. A very important 2-mark question again. 4 different types of viruses that are used in somatic cell therapy. The adenovirus vectors, adeno-associated virus vectors, retrovirus vectors, and herpes simplex virus vectors. So if you look into the slide, this gives you the advantages and disadvantages of using viruses as vector of different virus. If you see on the picture, there is plus and minus sign depicted under each virus. The plus is its advantage and minus is its drawback or demerit. So the first one, the first virus we see is adenovirus. It infects many cell types. So the advantage of these group of viruses, it infects many cell types. The disadvantage or drawback is it does not integrate into host genome, and of course it can be lost. So it does not integrate into host genome and can be lost. In that case, if you see, we require what is called as host genome to be infected to give a counterpart of the gene. The illena, of course, it is waste. So adenovirus, I repeat, the advantage, it infects many cell types, and the disadvantage does not integrate into host genome and can be lost. Regarding retrovirus, the advantage, if you see, it integrates into host genome and cannot be lost. It integrates in host genome and remains there. The negative counterpart is it can cause cancer. So they integrate into host genome and of course can cause cancer. So I repeat, retrovirus, the positive impact, if you see, it integrates into host genome and cannot be lost. And the drawback, it integrates into host genome and can cause cancer. Adeno-associated virus, designated as AAV, AA capitals, V capitals. The advantage, if you see, is integrates into host genome and cannot be lost, but it is difficult to work with. The DNA, complicated DNA, so it is difficult to work with. I repeat, the advantage of adeno-associated virus integrates into host genome and cannot be lost. It is difficult to work with is its drawback. And the next virus we have is herpes simplex virus, designated as capital HSV. The advantage, if you see, the DNA stays in nucleus without integrating into host genome. I repeat, the advantage, DNA stays in nucleus without integrating into host genome, and the negative impact, the drawback, if you see, or disadvantage is only infects cells of the nervous system, central nervous system. I repeat, vector advantages and disadvantages. Adenovirus infects many cell types, does not integrate into host genome and can be lost. Retrovirus integrates into host genome and cannot be lost, integrates into host genome and can cause cancer. Adeno-associated virus integrates into host genome and cannot be lost, and of course difficult to work with. Herpes simplex virus, DNA stays in nucleus without integrating into host genome, only infects cells of the nervous system. So this picture shows you gene transfer mechanism or gene therapy using an adenovirus vector. So on the top left, if you see, you have a vector molecule, the adenoviruses. Above that, if you see, you have a modified DNA to be injected into the vector molecule. You have on the either side the viral DNA, the top picture, top left picture I'm talking about. You have two sides viral DNA, and the center one that remains pink or reddish tinge is a new gene insert. So it's a new gene insert which is inserted into the adenoviral vector. And of course it is infected or it is transfected to an cell, maybe human cell. Vector is packed in vesicle. Vector breaks down, releasing the vector molecule. The vesicle breaks usually and releases the vector molecule that has a modified gene, and cell makes protein using the new gene. That is a picture that goes in an alignment. So the magnification is given on the top right, how it's attaching and injecting its DNA molecule. This comes with viral replication actually. Absorption, pinning, penetration, multiplication, replication, DNA release. That is what is its life cycle of virus, and that is how it also goes. And the counterpart of the virus molecule, that is in changed new gene, will make proteins that can be used by the cell in order to get replaced by the defective gene. So this picture is obtained from National Library of Medicine. What are the success rates till now for gene therapy? So gene therapy, if you see, is not exactly approved by Food and Drug Administration for sale. Even then, some diseases have been experimentally successful. Although no gene therapies have been approved by the FDA for sale, some diseases have been experimentally successful. The few examples of success rate, if you see, is melanoma, that is skin cancer, severe combined immunodeficiency, hereditary blindness, and of course sickle cell anemia. So I repeat, although no gene therapies have been approved by the FDA for sale, some diseases have been experimentally successful in treating skin cancer, melanoma, severe combined immunodeficiencies, hereditary blindness, and of course sickle cell anemia. And of course, few disappointments we have in gene therapy, why it is not yet approved by FDA, if you see. In 1999, a boy died due to an immune response to an adenovirus gene therapy vector. And of course, 4 children have adopted or developed cancer due to a retrovirus gene therapy vector. So commonly gene therapy methods, preferably if you see, we go in for what is called as germline therapy in order to control disease sequences for a young or an infant, for a baby. So there were disappointments for which FDA feels whether approval is to be given or not. The two reasoning, if you see, in 1999, a boy died due to an immune response to an adenovirus gene therapy vector, and 4 children have developed cancer due to a retrovirus gene therapy vector. So what are the ethical problems that raises because of gene therapy? Okay, gene therapy for serious genetic disorders or diseases, okay, but for other health problems, so all diseases, we cannot use gene therapy methods. Somatic cell treatment stays with the individual. Germline cell treatment, if you see, passes down the germline, which is, it is immortal. Imagine a situation, I can treat a mother egg with the normal form of gene, replace, you give a newborn who's with a healthy counterpart of gene. The same treatment can be used to create a criminal too, to change a person's disorder, mentality, and make him born, because all genes are physically expressed. So somatic cell treatment for treating disease of an individual, fine, but germline therapy, it passes to generations together, so it remains immortal. Are we going to select that? And of course, it is very, very costly, means it is not so theoretical as I was looking at the slides. It is not so theoretical, but to identify the genes, replace the gene, bringing out treatments is a very costly means. And who will pay and who is eligible, of course, is one ethical question, because any treatment procedure should touch a normal individual, should touch not only the rich, it should also reach a mediocre or below down to economically weaker sections. Economically weaker sections also it has to support, but since germline therapy or gene therapy technique is very, very costly, and who is going to pay and who remains eligible are the ethical question. I repeat, the ethical problem that we face using gene therapy for serious genetic diseases, fine, but for all other health problems, is no way possible. Somatic cell treatment stays with the individual. Germ cell treatment passes down the germline, so becomes immortal. It is very costly, and who is paying it, who is going to pay, and who is eligible for it. So this is one comic slide that you have. A person, would you say you had a dominant or recessive gene? He is asking the individual. Any doctor asks question to an individual for any disorder or any diseases he has as an history. So the similar way, a doctor is questioning a patient, who you say you have a dominant or recessive character, whether your father's or your mother's, so that it is inherited to you. So the question is, how far are we going to use gene therapy to treat individuals? Thank you.